Have you ever wondered why egg whites go from clear and gooey to white and rubbery when you fry them? No? Well, too bad. We're talking about it anyway. Welcome to the microscopic soap opera that is protein folding. And yes, there will be drama. Egg whites are mostly albumin, a protein that normally likes to mind its business with a neat 3D shape. But when you apply heat, all those delicate bonds snap like someone just spoiled a Game of Thrones episode. The heat waves cause bonds to break, which leads to hydrophobic parts jumping out and clinging together. Level 1, Primary Structure, aka the recipe. It's just a list of amino acids strung together like beads on a molecular friendship bracelet. This sequence is dictated by your DNA, which means if even one letter goes rogue, stuff gets weird. This is shown in sickle cell anemia, where the swapping of glutamic acid for valine and hemoglobin turns chill, donut shaped red blood cells into cranky, crescent shaped ones. Next is secondary structure, where things get twisty or foldy. This is the protein's Pinterest phase. You have alpha helix, which look like ribbon spirals and are held together by hydrogen bonds between parts of the protein chain, and beta sheets, flat paper fans, strands lined up together, held together by hydrogen bonds. Alpha helices are like protein curls, and beta sheets, imagine lasagna noodles made of science. In a beta plated sheet, two or more segments of a polypeptide chain line up next to each other, forming a sheet-like structure held together by hydrogen bonds. The hydrogen bonds form between carbonyl and amino groups of backbone, while the R groups extend above and below the plane of the sheet. The strands of a beta-plated sheet may be parallel, pointing in the same direction, meaning their M termini and C termini match up, or anti-parallel, pointing in the opposite directions, meaning that the N terminus of one strand is positioned next to the C terminus of another. But not all amino acids are team players when it comes to these chic structures. Proline, for example, is the drama queen. With its R group looped back to form a rigid ring, it bends the chain like a kink in a garden hose. That's why it's called a helix breaker. You won't catch it twirling gracefully in an alpha helix. Instead, proline prefers to hang out in bends and unstructured regions, keeping things flexible and unpredictable. Meanwhile, tryptophan, tyrosine, and phenylalanine bring big ring energy. Their bulky side chains make them awkward guests in tight helical spaces, so they opt for the beta pleated sheet, where there's plenty of elbow room to stretch out. Most proteins, though, aren't picky. They like a bit of both. So it's common to see a mix of alpha helices and beta sheets, with the occasional free spirited region in between. Others stay committed to just one structure, or go full rebel and skip both entirely. If a protein were a TV show, tertiary structure is where the plot thickens. This is the full 3D shape the polypeptide chain folds into, thanks to all the side character drama, literally, the R groups of the amino acids. These side chains are constantly mingling, flirting, feuding, and forming alliances through every kind of non-covalent interaction imaginable. You've got hydrogen bonds like the classic hugs, ionic bonds where opposites attract, dipole-dipole interactions like shy glances between polar types, and London dispersion forces, those blink and you'll miss it flings between nonpolar groups. Like charges, instant beef. Opposite charges, sparks fly. Hydrophobic groups, they ghost water and huddle in the center like introverts at a pool party. Hydrophilic groups, they work the crowd on the outside, charming water molecules like social butterflies. But wait, there's a twist. Disulfide bonds crash the non-covalent party with serious commitment. These are covalent bonds between two cysteines, acting like molecular safety pins to lock parts of the protein in place. Think of them as the protein's emergency backup clips. Once they snap shut, the shape isn't going anywhere. In the end, all these dramatic interactions shape a protein into the perfect 3D structure it needs to actually do stuff, like catalyze reactions or defend your body like a microscopic ninja. Some proteins like to fly solo. They've got their single polypeptide chain, their tidy tertiary structure, and they're happy doing their job alone. But others? They're team players, built from multiple polypeptide chains, called subunits, that assemble like a molecular boy band. In that team dynamic, that's called quaternary structure. Take hemoglobin, the Beyonce of group proteins. It's got four subunits, two alpha, two beta, working in perfect harmony to carry oxygen like a four-man relay team. Or DNA polymerase, a multitasking enzyme squad made of 10 subunits, all clocking in to build DNA strands like a molecular construction crew. How do these subunits stick together? The same soft skills that hold tertiary structures together, like hydrogen bonding, gentle handshakes, London dispersion forces, tiny nudges, and maybe a few ionic high fives here and there. It's less duct tape and more Velcro, strong enough to hold, but flexible enough to let the group adjust and work as one cohesive unit. So, while tertiary structure is the protein figuring out its own style, quaternary structure is protein social life, subunits collaborating to do big, bold, biological things. Proteins are drama queens with very specific taste, the right pH, temperature, and environment. Change the mood, too hot, too acidic, too harsh, and they fall apart. Literally. That beautiful 3D shape they need to function? Gone. Like a stressed out origami swan turned back into a crumpled napkin. This unraveling is called denaturation. The protein keeps its amino acid sequence, primary structure, but all its folding magic disappears, along with its function. Now, some proteins are chill and bounce back if you fix their environment. Others, once they're scrambled for life, like egg whites on a frying pan. No matter how gently you whisper apologies, that egg is never going raw again. But here's the plot twist. A few proteins can refold on their own in a test tube. No need to help. That means all the folding instructions are baked right into their amino acid sequence. Like IKEA furniture, but with better instructions. Still, most proteins aren't quite that independent and need help from chaperone proteins, aka molecular babysitters, that fold them correctly inside cells. So remember, primary equals sequence, secondary equals local folding, helices and sheets, tertiary equals full folding, quaternary equals subunits unite, denature equals sad, unfolded noodle. So next time you fry an egg, just remember, you're watching biochemistry in action. Delicious, gooey biochemistry.